But how does the parasite manipulate its host in order to ensure its own survival? Frederick Thomas, an expert on parasites, has studied the biological mechanisms of a nematomorph worm known as Paragordius tricuspitatus. These worms are rarely seen, as they live most of their lives within their insect hosts, usually a cricket. They only emerge when it's time for them to reproduce, which they have to do in water. Thomas decided to use this worm as a model to research how parasites function.
Most parasites are small compared to their hosts. Here it's easy to tell that we're dealing with a macroparasite, a very large parasite. Keeping relative proportions, it's like a human having a watering hose around 30 feet long in its stomach. It's a gigantic parasite that has grown from being a microscopic cyst to a very large worm, without damaging the cricket's internal organs, which it needs to keep alive until the end. And so it's only at the very end that it will change its host's behavior, forcing it basically to commit suicide by jumping into the water.
Thomas has studied these parasite-dictated suicides in swimming pools in the south of France. On some balmy summer evenings, he has recorded as many as 200 of these death plunges.
All species of nematomorph worms need water to reproduce. They're parasites which, as adults, are necessarily free to move about, and more importantly, are aquatic. So once they infect a terrestrial insect, they're obviously faced with a problem, which is how to get back to the water.
Before working its way out of the cricket's abdomen, the worms must first propel the insect into the pool, something crickets don't normally do.
In fact, if the cricket goes looking for water, it's not its own genes telling it what to do, but actually the genes of the parasite.
How can a worm, little more than a simple tube, possibly take control of a more advanced creature like an insect? Using the most sophisticated tools available to a molecular biologist, Thomas discovered that an infected cricket's brain produced twice as many neurons as that of a healthy cricket. And that's not all. One of our most surprising discoveries was the existence of apparently mimetic proteins, proteins that are produced by the worm, but which are structurally similar to those we know to belong to the insects. The worm has the ability to replicate the cricket's molecules, modify them, then send them back to the cricket. Following these forged instructions, the cricket begins behaving erratically, leaving its natural habitat and heading to the water. This ability to send molecular messages is not exclusive to this particular worm. Scientists have detected the same phenomenon in other parasites. Our work on the nematomorphs has made it possible for us to save a considerable amount of time on other areas of research. Research we're carrying out in connection with public health threats such as malaria or sleeping sickness transmitted by the tsetse fly and mosquitoes. Understanding the worm's tactics gives researchers insight into how other parasites manipulate their hosts.